IB Bio, Genetics Part 6 is a movie for HL students only. The movie will have its focus on 2 gene crosses, including the dihybrid cross. Near the end of the movie, polygenic inheritance will also be examined. The essential idea is genes may be linked or unlinked and are inherited accordingly. For clarity and emphasis, this movie examines unlinked genes. Here is the outline for all the movies available in the series on genetics. Use this outline to find the movie you need for review. This movie is focused here, and it's for HL students only. Here is the IB syllabus statement of interest. Calculate and predict the genotypic and phenotypic ratio of offspring of dihybrid crosses involving unlinked autosomal genes. The dihybrid cross is a cross following 2 unlinked genes, as can be seen here, gene A and gene B on different chromosomes. And the cross involves 2 individuals, both of whom are heterozygous for both genes. Di refers to 2 genes, hybrid refers to the heterozygous condition. And lastly, the crosses included in this movie involve unlinked genes on autosomes, genes on different homologous pairs. I'm using important language here. Pay close attention. Independent assortment is a very important concept in understanding dihybrid crosses. The law of independent assortment states that the alleles of a gene on one homologous pair pass to gametes independently of the alleles for a gene on different homologous pairs. Independent assortment of unlinked genes, like A and B here, is due to the random orientation of pairs of homologous chromosomes in meiosis I. Now, the possible gametes formed by someone, this individual, with this genotype, would be dominant A, dominant B for one gamete, recessive A, recessive B for the second gamete, dominant A, recessive B for the third gamete, or recessive A, dominant B for the fourth gamete. Now, if you're not sure about that, stay tuned, I'll explain, but I won't review all of meiosis. The expectation is that you have control of meiosis. Let's lead up to the dihybrid cross by starting with pure breeding parents. As pure breeding parents, each one produces only one gamete type. Each parent produces only one gamete type with respect to these 2 genes. You should understand why this gamete only has a single allele for each gene in the gamete. This is meiosis, which you need to bring forward. All the F1s of this cross, all the F1s are dihybrids, heterozygous for 2 genes. Red flower, full pod. So here is the F1, the dihybrid individual, heterozygous for 2 genes, and we're crossing the F1s among themselves. This is the dihybrid cross. 2 individuals, both heterozygous for 2 genes. Each individual in this cross, this individual and this one, will produce 4 gamete types. Think about independent assortment here. The next slide will help. Independent assortment of unlinked genes, like A and B here, is due to the random orientation of pairs of homologous chromosomes in meiosis I. During meiosis I, the homologous pairs align in either of 2 possible positions. We can have this lineup or this one. Notice the difference. Through anaphase I, when the homologous pairs separate, 4 possible gametes are formed with respect to these 2 genes. Dominant A, dominant B, recessive A, recessive B, dominant A, recessive B, or recessive A, dominant B. So here is the dihybrid cross, and here are the 4 gametes that each adult produces. And here is the Punnett square that accompanies the dihybrid cross. 16 boxes because each parent produces 4 gamete types. Random fertilization results in various genotypes. But before completing the Punnett square, it's important for me to say right now there are only 4 phenotypes, and here they are using the characteristics that I introduced earlier. 9 red full, 3 red wrinkled, 3 white full, 1 white wrinkled. Here's the completed Punnett square, and I've color-coded the 4 phenotypes. We have 9 red full, 3 red wrinkled, 3 white full, and 1 white wrinkled. The 9-3-3-1 is a classic ratio in genetics. Don't forget the 9-3-3-1 ratio when grappling with a dihybrid cross. It's a classic ratio because Mendel figured this out in the 1860s to produce his law of independent assortment. Now, you'll never be asked for the genotypic ratio. You will only be asked about the 9-3-3-1 phenotypic ratio for the dihybrid cross. So here's a 2-gene cross with an eye towards practicing the dihybrid cross. In Drosophila, gray body color is dominant to ebony body color, while long wings are dominant to vestigial wings. The genes for body color and wing structure are unlinked autosomal genes. Assume the parent individuals are homozygous for each gene. So the parent cross looks like this. E plus, that's the gray, VG, VG, that is the gray vestigial parent, crossed with the ebony, long-winged parent. Now in fruit flies, the symbols used are a bit different. The superscript plus denotes wild type, while the lack of the superscript denotes the mutant form. Work through the complexity of the symbols. Think carefully. It's not that difficult. Now notice, both parents are homozygous for each gene. They're homozygous for each gene. In other words, both parents are pure breeding, and we're going to work through the F1 and then to the F2, and it's the F2 that is the dihybrid cross. So here is the parental cross. Here's the gray vestigial wing parent crossed with the ebony long wing parent. Now, the gray vestigial wing parent only produces gametes that look like this. All the gametes have the gray vestigial alleles. This parent produces only these gametes. These are the ebony long wing alleles. Only these 2 alleles come from this individual, and all F1s are heterozygous for both genes. All the F1 flies are gray long wing. And we're going to mate the F1 flies among themselves for the dihybrid cross. The dihybrid cross sets up just like the last one that I exampled earlier in the movie. Each F1 parent, heterozygous for both genes, produces 4 gametes due to independent assortment. This individual has 4 gametes, this individual has 4 gametes, and the Punnett square is 16 boxes. You can stop the movie now to study this slide to ensure that you understand the 4 gametes that come from each of these parents. The expected phenotypic ratio, once fertilization has occurred, is 9-3-3-1. Remember, you'll never be asked for the genotypic ratio of a dihybrid cross. Just remember the 9-3-3-1 ratio. Again, you can turn the movie off now to understand where these 4 phenotypes come from in the Punnett square and understand why there are 9 gray long, 3 gray vestigial, 3 ebony long, and 1 ebony vestigial. Let's pause and step away from the dihybrid cross, but let's consider another 2-gene cross, but not the dihybrid cross. This individual is homozygous dominant for gene A and heterozygous for gene B, while this other individual, this other parent, is heterozygous for gene A and homozygous dominant for gene B. Now, before moving to the next slide, can you state the gamete types produced by each parent? This individual would produce 2 types of gametes, dominant A, dominant B together, and dominant A recessive B together. They're the only two gamete types that would come from this parent. From this parent, there are also only two gamete types, dominant A, dominant B together, and recessive A, dominant B. Those are the only two gamete types from this parent. Think carefully. You can stop the movie now and study this slide to understand the two different gamete types produced by this parent and the two gamete types produced by this parent. Here are the two parents from the last slide. I've attached some phenotypes just for clarity. In working through this cross, the Punnett square need only be 4 boxes. There are only 2 gamete types from this individual and 2 gamete types from this individual. Think carefully. And you can see the phenotypic results of this cross. The phenotypic results are that all of these individuals are brown and large. There are 4 different genotypes, take note, but phenotypically all are brown and large. So let's practice this idea of figuring out how many gametes an adult will produce and what those gametes might look like. The following genotypes represent unlinked autosomal genes. How many different gametes can be formed by each of the following adult diploid individuals? In the first individual, A, there are 4 gamete types, and they are A, B, dominant, dominant. They are dominant, recessive. They are recessive, dominant, and recessive, recessive. In the second individual, B, the 2 types of gametes, and they look like this only. In individual C, there's only 1 type of gamete, and looks like that. In individual D, as it turns out, there are only 2 types of gametes, and they are A, dominant, recessive, or recessive, dominant, recessive. They're the only 2 types. Think carefully, stop the movie if you need to. And lastly, individual E will produce 4 types. Maybe you can see it. It's not unlike the one at the top. We have dominant, dominant, recessive. We have dominant, recessive, recessive. We have recessive, dominant, recessive. And lastly, recessive, recessive, recessive. For the purposes of setting up a Punnett square to determine the expected ratios among offspring of 2 gene crosses, you must determine the number of different possible gametes for each adult in a genetic cross. Thus, how large would the Punnett square be for each of these following crosses? For the first cross, this first individual would produce 4 gametes, while the second individual would only produce 1 gamete, so the Punnett square would be a 4 by 1. In the second cross, this individual would produce 2 gametes, while the second individual, this individual, would also produce 2 gametes. So in the second case, it would be a 2 by 2 Punnett square. In the third cross, this individual would produce 4 gametes, and the partner would produce 2 gametes. So in this particular cross, the Punnett square would be a 4 by 2, 8 boxes total. In the last cross, the first individual produces only 1 gamete type, and the second individual also only produces 1 gamete type. It would be a 1 by 1 box. Now I do ask the question, could you work out the phenotypic ratio for the offspring of cross C? You can stop the movie now and practice, or follow along with me. Here's the cross. This individual produces 4 gamete types. This individual produces 2. We start to do random fertilization. We have this individual who is brown and large. We have this individual who is also brown and large. This individual who is also brown and large. And this individual also brown and large. Now there's 4, that's half of the offspring. We have this individual also brown large. We have 5 brown large phenotypes so far, and this individual also brown large. That's 6 brown large phenotypes. And lastly, we have, oh interesting, a black large animal, and we have a black large animals. So our phenotypic ratio is 6 brown large and 2 black large. Could you write the genotypic ratio? Here's a question for you to work out. It's a challenging question. 2 unlinked autosomal genes in dogs result in hair color and hair length. The allele for dark hair is dominant to the allele for white hair. The allele for short hair is dominant to the allele for long hair. Dark, short-haired dogs, okay, let's just write what we know. Dark, short-haired dogs are bred with dogs of a different phenotype, producing the following phenotypic ratio. We can see that here. And your job is to deduce the parental genotypes. Again, just to finish this out, to deduce the genotypes of both parents. Now we can look at the data and begin to make some assumptions. For example, we can see that there are 38 dark and 42 white. That's very nearly a 1-to-1 ratio. That should ring a bell for you. And if you look carefully, we have 60 short hair and 20 long hair. That's a 3-to-1 ratio. Be thoughtful here. So here's the cross, and this is the task to deduce the parental genotypes. I've pulled the two characteristics apart to show you an alternative strategy in dealing with 2 gene crosses. I have the data here. We see 38 dark animals and we see 42 white, and we know that that's a 1-to-1 ratio, and with that we can begin to answer the question. This individual is likely to be heterozygous, and then this individual is going to be homozygous recessive for hair color. Now, if we look at the other characteristic, which is hair length, we can see 60 animals that have short hair and 20 that have long hair, and that's a 3-to-1 ratio, and so we can see that here, and with that we can determine that this individual is heterozygous for short hair, and this individual also heterozygous for short hair. And with those 2 parents at the top, we can begin to put the gamete types in on the Punnett square, and what we're going to have is a Punnett square that looks something like this from that parent, and something like this from the other parent. And I'll let you do the random fertilization to determine the genotypic and phenotypic ratios of the offspring. It should match with the data here. Here's another problem. It's a 2-gene cross but not a dihybrid. In Mendel's work, he found that full pods were dominant to constricted, while round seeds are dominant to wrinkled seeds. In a 2-gene cross involving parents of unknown genotype and phenotype, the offspring shown below were obtained. Determine the genotypes and phenotypes of the parents and assume autosomal unlinked genes. Let's look at this by examining the cross of each gene alone. We can see that there are 6 full and 2 constricted. That is a 3-to-1 ratio. So with respect to pod shape, we can assume that this cross is heterozygous crossed with heterozygous. With respect to seed shape, we have 4 round and 4 wrinkled, and that would tell us that we have a heterozygous crossed with a homozygous recessive. And there are the genotypes of the 2 parents. If you're not sure, create the 8-box Punnett square to confirm the results seen here. With this slide, I have put one last 2-gene cross before you. Hopefully, everything seems very clear. This is a test cross, by the way. Do you remember the definition of a test cross? A test cross involves the breeding of a dominant trait individual with a recessive individual. Earlier, we used the test cross to determine an unknown allele. In this case, we know all the alleles of this individual. It's a dihybrid, 2 genes, both heterozygous. And this individual is the homozygous recessive, and by crossing with the homozygous recessive, we have a test cross. Now, throughout this movie, we have been following unlinked genes. In this case, A and B. A and B unlinked different chromosomes. And because of independent assortment, the dihybrid individual produces 4 gamete types. This should be review for you at this point. The homozygous recessive produces 1 gamete type. And here are the phenotypic results. Here are two IB syllabus statements that carry us toward the end of the movie. Describe two examples each of variation that is discrete or continuous. Explain how the phenotypes of polygenic characteristics show continuous variation. Discrete variation, seen here as one of two phenotypes, is typical of phenotypes controlled by only one or two genes. This graph clearly displays the discrete variation that results from a monohybrid cross where the probability of offspring is 3 red to 1 white. Here is a graph displaying a discrete 9:3:3:1 phenotypic ratio of the dihybrid cross. You can see the 4 discrete phenotypic classes of offspring. When many genes, 3, 4, 5, or more genes are responsible for the phenotypic character, the variation becomes continuous, as can be seen in these bell-shaped curves for height and birth weight. The bell-shaped curve displays continuous variation. Any biological character whose frequency appears as a bell-shaped curve is under the controls of many genes. Height, weight, intelligence, and more display continuous variation and are under the control of many genes, in other words, are polygenic. Here's the IB syllabus statement. Define polygenic inheritance. It is the additive effect of 3 or more genes, more than one locus, on a single phenotypic character. Polygenic characters display continuous variation, the bell-shaped curve. Now, polygenic characteristics are difficult to study because so many genes are involved. So there are some assumptions to keep in mind, that there's equal contribution of each gene, and that all alleles are equal, in other words, incomplete dominance. Here's an example of polygenic inheritance. We have 3 genes, 6 alleles, and this is polygenic inheritance with respect to skin color. For each gene, an allele for dark skin is incompletely dominant to an allele for light skin. All the alleles are equal. You can see the various combinations here, resulting in 7 different phenotypes, which is very close to the continuous variation seen in a bell-shaped curve. In truth, skin color is a polygenic character, but we do not know how many genes control skin color. Certainly there's more than 3. Once again, in this slide, we have 3 genes, we have 6 alleles. All the alleles contribute equally in an incomplete dominance way. We have 64 different genotypes, but there are 7 phenotypes if you look carefully at these combinations. This one has the same phenotype as this, as both individuals have 1 recessive and 5 dominants. These 2 individuals have the same phenotype, 4 dominants, 2 recessive, 4 dominants, 2 recessive, and so forth. Again, 64 genotypes, 7 phenotypes, again on the edge of showing continuous variation, the bell-shaped curve. So at this point, could you explain how the phenotypes of polygenic characteristics show continuous variation? You should think about the bell-shaped curve here. State that human height is a polygenic characteristic and is also influenced by environmental factors. List 2 environmental factors that would influence height. Environmental factors that influence height would require the person to be exposed during childhood when they're growing. Diet and exposure to certain types of chemicals or pollution during childhood could influence height. Once again, here is the bell-shaped curve that displays continuous variation of a phenotypic character controlled by many genes. Many human characters display a bell-shaped curve across the population. Human height, skin color, weight, intelligence, and many more are polygenic characters that display continuous variation. As a last note, and it might be of interest to you, we do have genes in the world that are temperature sensitive, allowing for animals to change color depending on the season. I'll leave the explanation for temperature-sensitive genes for you to research. You could add Siamese cats to your research. And that brings us to the end of IB Bio Genetics Part 6.